Okay, so back to references, which is where we laid off. So we have a read pool. It's obviously not a real read pool because we're only starting with 100,000 reads. We're down to 88,000 of those, but we can still align them to the reference. Pitfalls. Why do I call this a pitfall, right? Well, what's the biggest thing? The reference is always wrong. At a bare minimum, the reference genome from which your organism's reference genome was sequenced is not the one you have in the lab. Or if they're people, it's not the person that you're sequencing. It's a near neighbor, but there are at a bare minimum going to be SNPs, indels, mutations between the reference and what you have. It is highly likely that there are also whole regions that are misassembled by virtue of the fact that this is a computerized process and requires a lot of effort to actually finish a genome, right? Human genome is into revision 37, I believe, or 38. The mouse genome is on MM10 right now, which means that they declared it finished, and then they finished it again, and then they got to 6 and 7 and 8 and 9 and just started calling them what they are, which is releases of the genome. They're not right. They're just the best you have. So, what does that mean? The alignment then is also going to be less than perfect. On top of which, if you're dealing with a eukaryotic organism, you're dealing with introns, exons, splice variants, etc., all of which requires some sort of tolerance of aligning. This pipeline, if any of you are doing bacterial work, this pipeline is completely, from this point forward, overkill for what you'd be doing. Top Hat is built to be tolerant to introns and exons, does a lot of extra work that you don't need. If you're going to do it with bacteria, you can just use Bowtie 2 or BWA, skip the next two steps, and just go to the counting. I'm going to talk through what this is, and I'm going to have you copy and paste and run it. So, Top Hat is going to run with a bunch of options. You'll see that it's got here genes.etf, the transcriptome index home, the name of the output directory, and then the name of the reference genome, and then your input files. Go ahead and copy and paste and get that started. What is it actually doing here? It's trying to do alignment. What is it aligning and what is it aligning to? Why is it using... So read aligners. How many people actually know what a read aligner is and is doing? So read alignment is the process of taking a reference sequence, taking short reads, and getting them to actually display the point at which they should match to the reference genome. It takes a lot of work. It takes taking the reference genome and indexing it for whatever tool you're using. It takes copying or finding a way to search through that information to be able to figure out what the location is. And then on top of it, because we're trying to do transcriptomics, it has to be tolerant to the alignments and gaps, so it has to be able to just align part of the read and part of the read somewhere else and record all that information. If you were doing this to a bacteria with no introns and exons, you would just align it. All you would need would be the directory of the genome, and everything else you could just say, I don't care about it, and I'm going to see what happens. We need the genes.gtf file. What is genes.gtf? It's a representation of the gene calls for your reference organism. So it says, this gene occurs and appears at this region of the reference genome. The transcriptome says not only that, but these are the introns and exons of that. So what we're doing is we're taking this and the reference genome and building a transcriptome index, and then aligning these two read sets to it at an output directory. Does that make sense? Are we kind of okay on this? This is the step when I told you that if we were going to try to do this with the full 150 million reads, we would end up waiting 4 hours for it to finish. As it is, this is probably going to take a good 10 minutes or so. Well, not 10 minutes, a couple minutes. Let's see. The largest portion of what it's going to be doing here is the header and then the indexing. all of this work that it's doing is work that it is doing to the reference genome. It is preparing the reference genome for the alignment, which is going to take much longer for us than the actual alignment. For a normal set of reads, it's going to be trivial in comparison to the amount of time it's taking to do the actual alignment. Right? We're taking 100th or a 500th or 1,000th of the actual data set for this to run. So this is going to take a while. We're going to let it sit up there while I talk about other things. Sort of. Okay. So the next steps require that that step finishes, which means that it's a really good time for you to ask me questions. Questions?
So back to those first arguments that we just actually
For Top Hat?
Yeah, for Top Hat. We just have bacteria, but you want to do
Top Hat 2, like I said, is a tool that is built for reference genomes with complicated transcripts, with introns, exons, etc. Bacteria don't do splicing, so you don't have to worry about it. So all you have to do is actually align the reads to the reference genome. All that, so we'll get to it in a couple minutes when we talk about what HT-seq does, which is the pipeline that I've got here. All it needs is the location of the alignment and the actual names of the genes in the genome. So to do that, you would use a much more simple aligner. So BWA or Bowtie would be the tools, you've probably seen them before, that you would use instead. And then instead of all of this, it's just Bowtie 2 reference genome output file reads. So let's, here, I think I can probably pop that up relatively easily. No, this is probably not the best way to do it. I'm trying to figure out the best way to show you what it's doing here. So the alignment itself is occurring to the reference genome. Right? The reference genome is starting at base 1 and going all the way through, if you have multiple chromosomes, through each chromosome. If you have one circular chromosome, through base 0 and base -1, where they join together, and prints that whole reference, the whole chromosome. So the reads are actually aligning to that. What Top Hat is doing, when it's using the genes and the transcriptome bits, is it's also trying to become, or to maintain awareness of the introns and exons that are listed in genes.gtf. Genes.gtf is an annotation file, so it says the gene exists at these locations. If you have a bacterium, the gene starts here and ends here, and all you need to know when you're doing your analysis downstream is what the name of the gene is and what reads align within that region. What Top Hat needs is to be able to do that, and also, by the way, say, all of these places where it splits apart, I need to know what those are. So that model is in the GTF file if it's necessary. When you do an alignment to a bacteria, don't need any of that. All you needed to do is just align to the reference genome, and then the tool downstream is going to use the GTF file to give you the names of the genes and the locations. Top Hat set, so it made a new directory. I'm also in a different file system. So it made a new directory for everything that we did. So we're going to go look at it and make sure that I have all of this. So you'll be a little behind me, which is fine.
Yeah,
Well,
we're probably all doing it at the same time.
and it's doing work in a funny way, so that's fine. It may, in fact, not work out for everyone. It should, but well, we're not going to worry about it. So the first thing we're going to do is, in that directory, what do we have? We made a whole bunch of files. There's the summary.txt file, which tells you what it did, acceptedhits.bam, which is probably the most important, and then a bunch of bed files, some info, and the logs directory. Accepted hits is really the only important bit that we care about in this for now. If you're curious about how the alignment worked, what it threw away, anything like that, you can read through the alignment summary, but we're not going to worry about that. What we are going to do, see if this So work as advertised. We're going to count how many reads were accepted. SAM tools is the tool set that deals with BAM files, SAM and BAM files. We're going to view it using the -C, which is to count, using capital F4, which is saying count only reads that have a valid match of my BAM file. So, 174,283 hits, more or less. What does that mean?
Number of reads that were aligned.
It's the number of reads that were aligned and accepted is aligned to the reference genome. Is that a good number? Is that a bad number? We can actually tell. I'm not asking because I don't know the answer. I know what the answer is. I've done this before. So how many reads did we start with? About 88,000 times 2, right? That was what we had after trimming. So we had about 176,000 reads. We have 174,000 of them aligning to the reference genome. Is that good? It's great. Should we be concerned? Is it too many? So what would make it be lower? What kinds of things could cause the read alignment value to be lower than 90%? When should you be concerned about it? Go ahead. So quality starts to drop below 90. Are you sure that your reference genome is good enough that the stuff that isn't aligning is not aligning because it's... So what does it mean if a read that you got out of your RNA sequence experiment didn't align to the reference genome? What does that actually mean? It means that the read is not present in the reference genome. Should that be something that you're concerned about? If it starts to drop really low, did somehow you get your RNA sequence experiment contaminated with something else? That would be really bad, right? That would be a real problem and you should go investigate it. On the other hand, if you're dealing with an imperfect reference, if there are whole regions and gaps of your chromosome that's missing and that happens to be a highly expressed gene in your condition, you won't be able to tell the difference, right? So when you start dropping down below 90%, something could be wrong, or it could just be that your reference is bad, or it could be that you've discovered a new gene that hasn't been finished yet. All of these are options, but with a rule of thumb, anyway, is the way that I would say it, is that 90% is a good cutoff to think that you have good sequence data for a model organism. 75, 70, 65 is probably okay for a semi-model, depending on how much work has been thrown into it, because there's going to be a lot of reads that don't exist there. And if you're doing this with an organism without a reference, you're probably assembling de novo, so this doesn't mean anything, right? So it's going to be a different pathway. That's actually decent, and it could, in fact, be that you're getting multiple reference alignments, the same read, so we're okay. We're not unhappy with that number. So what do we do now? We have now aligned the reads to the reference, for those of you that haven't finished yet. We're going to pretend we have anyway. And then we're going to copy in this next set of commands, which is using a tool called HTSeq. So PySam is necessary for HTSeq to work. I don't think this line is actually necessary anymore on the HTSeq, but we're putting it there anyway. And we're going to actually perform the HTSeq count command. So, I don't like it on this, so I'm going to go back over the web page. It's taking an input file that's in BAM format. In this case, because this experiment is old, the data is not stranded. Or at least this says it's not stranded. The new stuff, right, depending on how you sequence it, you're either going to have strand-specific RNA sequencing or not. You want to throw that flag there. Order, what's POS? I do remember what POS means on the top of my head. Probably positional. Oh, right, because the BAM file has been sorted by the reference genome position. So when you run top hat, it sorts the output based on the reference genome. So it's positional sorting rather than read number sorting. And then your input file, and then your GTF file again, and then an output. So I'm going to hit return because it doesn't take that long, and then I will So I'm actually running the commands there, and it's doing a count. So what it's going to do is take all this information that we made in the BAM file, plus the genes file, and it's going to turn that into a count. So the output here is going to be the gene name, a tab, and the number of reads that align to that gene for every gene in your reference organism. So we're getting close, right? We know that there's about 100,000, GFF lines. So this is having to process through the reference genome. So it's going through every line of the reference genome. We're at 1 million. So showing you a SAM output. So this is one line of a SAM file or BAM file. The name of the read, which again is an incredibly long, useless name that is always there. A bunch of information about what kind of alignment it was. Information about the position and chromosome of the alignment. How long the read was. How good the alignment was. Position. So position in the reference genome, or position at the end of the alignment in the reference genome. And then the sequence, the quality, and a bunch of stuff. So it's taking all of this information every time you do an alignment, and it's outputting it to a file. And then it's taking that information and trying to get the actual genes that that's aligned to. We are right now, while it's running, taking a sidetrack from there are a bunch of different pipelines you can take for RNA sequencing analysis. The one that you will find if you go to the Top Hat webpage is to use bow tie, top hat, cufflinks, cuff diff, cummerbund, etc. to analyze your data. There's not necessarily anything wrong with that pathway. I'm choosing a different pathway. The reason why I'm choosing this pathway is because about a year ago, Titus put out a call to our friends on Twitter and said, which tool do you think is the best tool which pipeline do you think is the best one to use for RNA-seq analysis? And the general consensus was to use Top Hat into HT-seq into HR. That is based entirely on recommendations of other people who are not you. If you don't like this pathway, you are welcome, or pipeline, you are welcome to use a different one. They will give you slightly different results. Before I go forward with looking at what these results are, what should we do when we have slightly different results? Right? This is another question. If you're going to run an analysis, somebody at some point is going to say, well, why don't you use a different tool to do the same analysis and then see which results make more sense, or do something with both sets of results? What do you do? Right? So this is a question, and I'm putting this to all of you because most of you are the person in your lab that your PI is going to come to and say, do this thing. And so it is your job to think about how much work that means for you, how much value that gives, and what that actually is going to mean. So we're going to take all this stuff and go through EdgeR and get a list of differentially expressed genes. If we wanted to do the exact same thing and take the counts and put it through DE-seq, which is another package, we might get a slightly different set of genes. If I have a slightly different set of genes expressed by two different pipelines, what would you do with those two pieces of information? How? So combine them is a statement. Do you combine them by taking only the ones that are true in both? Do you take all of both and keep them? Do you keep only the ones you like? What do you do, right? And the reason I point this out is that the biggest danger you can have in RNA-seq with high throughput anything, with large data sets, is for people to go expectation mining, right? Confirmation bias in reverse, if you will. I don't see the gene that I expect to be present. Redo the analysis until you find the gene. bad idea, right? You need to find a pipeline that is going to work and give you results that you trust, and if the expected result is not going to match what your outputs are, then you need to either determine whether or not you're right or wrong before you start looking at a different tool. I'm not going to tell you not to use multiple tools. I'm going to tell you that as soon as you use more than one tool or more than one pipeline, you're opening yourself up to a world of confusion, and you need to plan in advance what you're going to do with it. You can combine the results. You can do any of those options that I just mentioned. I just don't know which one's right. And I'm not sure that anyone will. So make sure that when somebody comes to you with I just found this new tool in a paper, maybe you should run it too. Be aware that you're the expert in the room now because you've been in here for 2.5 hours, and you can tell them that they're wrong. Please do. Make sure that you don't do work that you don't need to before you get to results. Okay, so that all being said, what do we do? Well, we ran a command, and that little greater than sign at the end of the command here was taking all of that output and putting it into a new file called salivary rep1 counts, which we happen to see, where's counts? Let's look at it. Easy. Everybody has data. You can show this to anyone you want, and they'll be able to run with it, right? You know what this means? I don't know what this means. I mean, I know what it means. It means that there's a gene with an ensemble ID of this that has no counts in my sample. But what does that mean? Well, now we're back to the final point that we have to talk about in RNA-seq, is that you can't do RNA-seq in a vacuum, right? You can't do RNA-seq analysis on a single sample. So we did all of this work with 100,000 reads from the salivary rep1 sample set. Can't do anything with it, though, so we need more data. So next, we're going to learn a little bit about bash scripting. So if you see here, we're going to go back to the root directory, and we're going to create a new file that we're going to call sample2. So go ahead and copy paste. So for those of you that have been able to do this, if you copy and paste the code, you should end up seeing a box that looks something like this. What we've done is we've used a file editor called Nano. It's the simplest text editor you can have on our HPC. It's a what you see is what you get type program, and then you can type control things to do other things. So when you want to quit, you hit control X, and it'll ask you if you want to save the changes. You can say yes and hit return, and it'll ask you what file name you want to save it to, and you hit return again, and you'll get back out to the screen. In the meantime, I'm going to copy and paste this in, and we're going to look at it while we're waiting. So the first line of any program on the HPC on a Linux system, for those of you that don't know, needs to start with the kind of command that you're going to run. So in this case, we're doing what's called bash. The stuff that we've been typing or copying and pasting line by line as a bash command that does things. So really this is, or should be, an exact copy of what you would type by hand if you were going to do it manually. Going back to what I said loudly and vehemently at the beginning of this whole section, replication is important. The ability to do the same thing again and have the same output come out is really, really important. Typing things on the command line is a really good way to not be able to replicate your work ever again. So the first step to that part of it is to try to do all of your analysis in some sort of script file so that all of the commands are right there. When you need to change them, you have to change the file itself and run the whole program again instead of typing a slightly different command. It's also a really easy way to be able to change a couple of things and say use this exact same template for a different file, for a different input set. So, what is it going to do? It's going to do a bunch of things that we already did. It's going to load modules, which is where all the software is using, FAQCS, FastQC, Trimmomatic, Top Hat, HTC. And then we're going to do things. So it's going to load all those, change directories into the RNA-seq model, make dir QA, which you might remember, and it's going to take those heads of the next data set. So before we were doing the saliva rep 1, now we're going to do lung rep 1, right? So the input of those is 15.326, we want it to. So it's going to take those, and then it's going to change into QA directory, do the FastQC, do the FAQCS and the Trimmomatic, and then the top hat, and then the counts. Right? So it's actually not a very long program. Everything that we've done over the last 2.5 hours has been 5 commands, 6 commands. But now we know why we're doing it. So if we want to move forward, we can keep moving forward and do the same thing again. So now that I've copied everything and we've talked about it, you type Control X to exit, and it asks, do you want to save what you wrote? Say yes. We really want to save it to sample 2, which is what we started with. So now we have a file that we made called sample2.sh. To run that, you type bash and then the name of the program, and it's going to do all of that work. And if you saw it said make dir, cannot make directory if you saw it before it scrolled off the screen. So feel free to go ahead and let it start running, and we will just kind of sit and wait and talk about things while we're letting it go. All right. Yay, I have data back. I got data, right? Everybody's happy? Well, you probably don't have data yet because I started before you. So in the QA directory, I now have salivary and lung rep 1. So the same way I did the SAM tools count for salivary, I can do the same thing for lung. So here we come to our first conundrum. Right? For those of you in the back of the room, sorry. I did my lung counts, and my lung counts are significantly lower than my salivary counts. Is that a good thing or a bad thing? Right? What do we do here when we were looking at it? So what we don't know, because we automated all of this, is how many reads we actually trimmed. Sorry, I just lost my connection, so I may not be able to talk as much as I wanted to. So, right, I did the same thing that I did before. I was curious. I wanted to see how many of my reads align. Well, this time I only have 133,000 reads aligning. Is it a bad thing or a good thing? Why is it good? The point being that there's only 133,000 reads out of the original 200,000 that align. But that's because we trimmed off an extra 30,000 reads out of the lung sample than we did out of the salivary sample. Right? So if you look at what we had in FastQC here, we've got 72,000 reads that survived after all of that, which is significantly lower than what we had in the salivary sample. So if you're going to have any concerns, you wouldn't have concerns. The data still look good, right? The number of the surviving reads 72,000, of the 72,000 surviving reads, 90+% aligned to the genome. So that's great. If you want to have concern about why 30% of these reads got trimmed out versus 15% out of the salivary sample, that might be something that you could take and be concerned about. But that's a different question, right? If we assume that that's just something with the sequencing run, we'll move on with our lives. We'll say, well, look, now we're good because both of those came out looking more or less similar. We could, if we wanted to, at this point, go forward and analyze 2 samples, 1 sample each, 2 conditions, and edge-R. I don't really want to because we have 2 more samples that we can work with. The next bit and piece in this is to try to see if we can make a new version of sample 2 that looks like the sample 3. Well, what I'm going to do, and you can copy this, is not on the web page, so you can so I'm going to make a copy of sample 2 and call it sample 3. And I am going to edit that 26. Oops. Oops, not 2, 3. The same way, right? So all I did was I made a copy of the file, and then I typed nano of the new version of the file, which is now called sample 3. So, what do I need to change to do the same analysis with one of the other samples? So when I'm going to change my input FASTQ file. Oops.
25.
Well, 25 was the first one we did.
Oh.
There we go. So I'm going to replace that with this. So I happen to know, or at least think I know, that 82 is the other salivary sample. So I'm going to change that. That fixes all our problems, right? Whole file's good to go? No? You sure? Fine. Okay, so now we have to change all of this, right? So the input files here are no longer lung REPL 1 and lung REPL 2, but see what I'm doing here? Line by line, I'm having to go through and change all of the file, all of the names to make something new. I'm going to keep working at this. I would recommend you all do the same thing. And if you want to get really fancy and type faster than I can, you can work on lung rep 2 as well.